And the exciting thing here is that we are a British listed company that is leading the world in cancer diagnostics. And I'll explain to you why I believe that is the case. First of all, cancer is a very pervasive disease now. And if you look at the National Cancer Institute, United States, an estimated 40% of men and women will be diagnosed with cancer during their lifetime. Our mission is to enable personalized cancer care with a simple blood test to guide treatment. And this can improve patient outcomes and reduce healthcare costs. So it's a double benefit. And we can do that by recovering cancer cells from blood for analysis, even though they're very rare. There may be only one cancer cell in amongst 1 billion blood cells. We have pursued over a sustained period of time an Fda Product Clearance. So Fda is the US regulatory system, and it is considered to be the global gold standard. We successfully announced in May of this year that we had achieved that goal, and we were the first, and we are the only company in the world to have achieved an Fda clearance for a device to harvest cancer cells from patient blood for subsequent analysis. This was a huge effort, 6 years of work, 15,000 samples, and 400 technical reports, and it gives us a very significant market lead over others who wish to address this market. As far as we're aware, having asked the FDA, there are no other companies even started such a process. And the key to this is that the Parsortix system can address flaws in the standard of care in patient management. If you look at the National Cancer Guidelines in the United States, they require a biopsy of secondary cancer sites in breast cancer. So what that means is if the patient's got breast cancer and it's spread to another organ, such as the lungs, the liver, or the brain or the bones, then the physician should seek to do a tissue biopsy of that secondary site. Well, as you can imagine, it's very difficult to cut out tissue from the lung or tissue from the liver, or even worse, trying to get tissue from the brain. In fact, that's so dangerous they won't do it. So despite it being in the guidelines that all patients should have such a secondary tissue biopsy, it only is successful for around 50% of the patients. The other ones fail to get it because they're too sick to withstand an operation, or because there's insufficient tissue available. The reason that the cancer guidelines call for such a biopsy is that cancer changes over time, so that the initial information that the physician gains from the tissue biopsy on presentation is out of date. And doctors are actually flying blind in their treatment of patients. They don't have up-to-date information on what the cancer is doing in that particular patient at that time. So we pioneered an approach to get those cancer cells from a blood test as opposed to the invasive tissue biopsy. And we do that with the Parsortix instrument, which is the machine shown on the left-hand side of this slide, which is coupled with a one-time use Parsortix cassette, which is held between finger and thumb, top right of that slide. So that is a razor blade model. You use a new cassette for each blood sample that you process. And this approach that we've developed is patented worldwide, it's automated, and it's simple to use. I'm going to show a quick video to show how that works. What you're looking at here is a Parsortix cassette with an inlet, and the blood flows down the channels that are shown here. And as you zoom in on those channels, what you see is a series of steps. So the red and white blood cells can go up that staircase, and then they go through the critical gap at the top here and flow away. But the cancer cells, which are much larger, they get held gently at that critical gap, and they don't make it through the gap. And in that way, we separate the blood components of red and white blood cells and plasma liquid from the cancer cells which we want. And the machine will do all this automated on an automated basis, and then will reverse flow and recover the cells for analysis. Now, this video shows this happening in real time for a breast cancer patient's blood, and this is 100 times magnification under a microscope. And what you see there as those curved lines are the edges of the steps looking down from on top. The deep red color is the incoming blood. The light color here is the single cells going through the critical gap. and then this is the exiting channel as the blood components flow away. So in a minute, you're about to see a breast cancer cell. Here it is, and it's on the final step. So it doesn't make it through that gap. All the remaining cells flow either side of it, and you end up with the target cancer cells recovered, even if there's only one cancer cell in 1,000 million blood cells, reliably and effectively recovered in our system. And as I mentioned, the machine will then automate the process of reverse flow to recover those cells. So what does it actually mean if you can get hold of cancer cells from a simple blood test? Well, first of all, those are the cancer cells that are actually spreading the disease within the patient. And over 90% of patients who die from cancer die from the metastatic spread via these cancer cells, which leave the primary tumour, go into the blood circulation system, and then flow around in the body and land somewhere else and grow into a secondary cancer. And that's what ultimately will kill the patient if they're going to die. So if you can reliably get these cancer cells, you have a real-time, up-to-date sample of what's happening in the patient from the cancer's point of view. And you can use that to detect cancer in high-risk groups. You can use that to interrogate the cancer cells to work out which drug would be most effective. You can assess whether treatment is going well or not. And importantly, you can use it for remission monitoring. If a patient is in remission, they should not have these cancer cells present in their bloodstream. If, on the other hand, they get those cancer cells in their bloodstream, it is demonstrating a risk of recurrence or relapse. It does not mean they have relapsed. It means that the cancer cells are present and they may be about to relapse. So it gives an opportunity to treat the patient ahead of such relapse. And longer term, it can be used in early cancer detection in wider deployment. So all of these different activities can be done without modification to our Parsortix system in every different cancer type that it's been tried in, which is now 25 different cancer types. So that gives you a market opportunity of $100 billion US. So circulating tumor cells, which are these intact living cancer cells that the Parsortix system recovers, provide you with an up-to-date, real-time information with a complete picture on what's happening in the cancer, DNA, RNA, and protein expression, all of which are very important. And that's exactly comparable to the tissue. So tissue biopsy, for example, in breast cancer, they cut out part of the tumor and take those cells to analyze in a laboratory. That's what we mean by tissue biopsy. The problem with, you can do all the DNA, RNA, and protein expression with tissue biopsy, but the problem is you can't repeat it, because once it's been cut out, it can't be cut out a second time. So thinking breast cancer, you will find that the patient will have had a lumpectomy or a mastectomy on presentation, and therefore there is no tissue to access unless and until it spreads to a secondary organ, which, as I've explained earlier, is very difficult to access. So the circulating tumor cells are the best sample to give you up-to-date information on what's happening in the cancer. There is and this whole process is called liquid biopsy. So we've got tissue biopsy, which is looking at solid tissue, and liquid biopsy, which is recovering cancer cells from the bloodstream, which is a liquid, and therefore liquid biopsy. The industry is by and large focused on an alternative liquid biopsy, which is called circulating tumor DNA. And that's fragments of dead cells, which is present in the blood only for a short time prior to excretion. Actually, this is a very limited analyte because it only allows the measurement of DNA, and it doesn't allow the measurement of RNA and protein. Nevertheless, Angle has pioneered the way to work alongside of the ctDNA companies by doing ctDNA and CTCs from the same sample. This approach that we've developed has now been in the market with leading cancer research centres utilising it for several years. And in fact, we've got 33 independent cancer centres now who have published peer-reviewed publications totaling 70 in number, looking at 25 different cancer types. These publications are freely available on Angle's website, should you like to review them, and a clear demonstration of the evidence of capability of our system. We started work in breast cancer, and that's where our FDA clearance is. We've moved into ovarian cancer, where we announced some superb results on ovarian cancer detection this morning. And we're soon moving into prostate cancer. And as I mentioned, there are 25 different cancer types that can be addressed. We've got a multi-pronged commercialization strategy. We've got a pharma services business where pharmaceutical companies pay us to analyze the blood of patients before, during, and after their trials. We've got a partnership program where we're working with large medtech companies seeking to combine their downstream technologies with our ability to recover the cancer cells to increase their revenues. And we sell the product widely, and we're building out a distribution database to deliver that. The pharma services business is now well established. We've got 4 established customers in place, and we've got 20 further biopharma companies in the pipeline, including 3 household names, any one of which could provide very large-scale revenue opportunities in its own right. We charge $1,000 or more for each sample that's processed. It's a $2 billion market in just these 4 cancers. In terms of the results that we've announced this morning in ovarian cancer, we've just completed a 144 patient study in a real-world environment to detect the presence of ovarian cancer in women with a diagnosed pelvic mass. So women with an abnormal pelvic mass require surgery. If they have cancer, they need specialist surgery and other treatment. If they don't, then they can stay in their local hospital. We've achieved best-in-class performance with sensitivity and specificity above 90%, which far outperforms current tests in the market for ovarian cancer, and enables a significant reduction in false positives and false negatives. We've signed a deal this year with a big group called Solaris Health to do prostate cancer studies, and we expect to launch prostate cancer studies or complete our studies by the end of next year. So we're in a very strong position at Angle. With the FDA clearance is a major breakthrough, and it's bringing us global recognition. We've got first mover advantage in a huge market, and we're now in the position to capitalize with multiple corporate partnerships with leading medtech companies and pharma companies. I'll stop now because I want to make sure that there's enough time for questions. Can we go to questions now, please? And can you bring Ian into that as well, please?
Angle collaborated with Abbott in the metastatic breast cancer trial that led to the FDA approval. Now that you've got approval, how do you see your relationship with Abbott developing?
Yes, well, they're the leading player for HER2 testing. It's currently a $600 million market, and they're the largest player in that market. So we utilize their downstream path vision test for HER2 in our FDA submission. And we're now in detailed discussions with Abbott about how we can work with them to bring forward a test combining Parsortix and path vision. And those conversations are progressing very well.
Excellent. Okay. Is the Parsortix cassette, is that IP protected?
Yes. So we've got 26 granted patents so far across various jurisdictions, but critically in the US and in Europe.
Okay, thank you, Ian. Well, back in July, you raised 20 million. How was that invested?
Well, obviously, we have planned expenditure, planned investment in various areas of the business. So there's continuing to develop the pharma services side of the business. There's building out the launch of the PC1s, which is the FDA approved instrument, and establishing distributor networks. So a lot of the money is going towards building our sales and marketing and commercialization aspects. There's the ongoing R&D activities, so bringing in other downstream analysis types, and there's progressing in particular the prostate cancer study with Solaris, and building out the opportunity in the ovarian cancer. So we're deploying the capital across core areas. Obviously, we've got a few major business development areas that we're progressing. We've got a finite amount of capital, so we're going to keep that tightly focused. But we're delivering on the core areas, and investors backed us to continue to progress the business.
Thank you, Ian. With FDA approval, why has the share price not done a great deal?
Ian, do you want to answer that?
Yeah, I mean, obviously, the share price is what it is. We've we've continued to deliver on our milestones. There wasn't the sustained response on the share price that we'd have hoped for. It went up and then fell down fairly quickly. Obviously, the broader macro drop is that performance for growth companies has been challenging, but all we can do is focus on our milestones and our delivery and continue to progress against that.
Okay. When do you forecast a break-even situation based on forecast future revenues?
So
yes.
Shall I start and then you can pass on?
Okay.
So, I mean, this is a strategic question for Angle because we already have profitable businesses. Our pharma services business is a profitable business. We get paid $1,000 or more for a sample that comes in, and we can process that at around about $100. So we've got a good profitable business in pharma, which is available to us now and growing fast. We also have a product-based business which has high margins, and we make money on everything that we sell. So the question is, to what extent do we want to continue on a land grab policy of investing in new cancer types and new applications versus commercializing what we have now? And that is a strategic decision, because there's a lot of the expenditure that the company has is discretionary expenditure designed to grow the value of the business. Ian, do you want to make
and as Andrew said, we're running multiple areas, and this includes investing in significant studies. So that includes what we've previously done on the ovarian cancer, just better into the results today. It's investing in the studies for the prostate cancer. So we've got particular areas of the business we're investing in, and that's to generate revenues further down the line. But there is also that focus on the nearer term commercial revenue. So overall, we follow this sort of tightly focused plan in the breast cancer, in the ovarian, and the prostate, and we're targeting break even in 2020. That's cash flow break even in 2025.
Thank you. Does Angle intend to publish the sensitivity and specificity data from the metastatic breast cancer trial that led to FDA approval?
Well, it wasn't a trial which was measuring sensitivity and specificity. What it was a whole series of trials actually to prove the performance of the instrument in recovering cancer cells. So it wasn't as if we were diagnosing cancer. What we're seeking to do is to recover cancer cells in known patients for subsequent analysis. However, to give a bit more flavour on that, as will have been seen in the announcement this morning, we've secured a sensitivity of 90% and a specificity of 93%, which is way higher than anyone else has ever done in the detection of ovarian cancer in a real world, very difficult to analyze sample. So basically it was women with a known pelvic mass, whether that was benign or malignant. So that gives you an indication. In terms of the performance in breast cancer, we know that the Parsortix system reliably recovers cancer cells, generally between about 75 and 85% of cancer cells which are present. So not all of them, but the vast majority. And the issue here is just as a tissue biopsy doesn't need to cut out all of the cancer to be useful, the same thing with our technology. What we need to do is to get at least one cell so that that can be analyzed. Well, as we know, we've had some very good results, which led to the first ever FDA clearance in that regard.
Yeah, and if I can just add to that, as I said, it is an FDA clearance in metastatic breast cancer. It is a platform clearance, so the data and everything that was generated was to support that. Obviously, we worked with 4 of the leading cancer centers in the US, and we expect multiple publications to come out of this, both reflecting their work, but also, we ran overall more than 16,000 samples were processed for various things. So we'll also have plenty of internal publications. Now, there may be some areas of the work that we did that we don't want to publish and keep it as a magic mix. But yes, we've got multiple publications planned to come out, and that'll be a mix of timings.
Okay, I've got time to squeeze in 2 more, I think. Are national hospitals, health systems showing interest in the Parsortix system? Yes,
and I'm pleased to say that we're beginning to get engaged in the UK as well as the United States and Germany. So that's a good step. So at the moment we are offering the Parsortix system as an capability to recover cancer cells from blood for analysis, but we don't currently offer any way ourselves to analyze those cells. So there is a bit of an onus on the customer to be prepared to work up how do they analyze the cells and what does it mean. So that is a customer responsibility at present. Soon we will be launching some downstream analytical kits to go with the cells to make it easier for the customer to adopt. And that's all part of our product development program.
Okay, time for one more. In order to execute your plan, how much cash will you need to raise in the next 3 years?
Yeah, so keeping to the tightly controlled focus that we've got, then it'll probably be anywhere in the region of $30 to $50 million. Now, that doesn't mean that's money that has to be raised from the fully from the markets. Obviously, we're working with large corporates, and we'll be seeking to bring in some non-diluted funding from partnership deals as well. So there's a balance here. What we've done with the funding that we raised recently in July, that was designed along with existing cash, R&D tax credits, and sales. That was designed to take us out to June 24. So we're in a good position cash-wise, and what we'll be seeking to do is take the business forward as smartly as we can on the commercialization side, but it would require some additional capital. The question is, what source will that be from?
Yeah, and again, just to my earlier point, this is all about strategic decision making by the company. So our pharma services business is fully funded and paid for by pharmaceutical companies who pay us to develop assays that they want, who pay us to process samples in their clinical trials, and ultimately will pay us to provide them with companion diagnostics in clinical use. So all of that is paid for by pharma. It doesn't cost us anything. They pay for all of it. So we could decide today to curtail other activities and focus in on just pharma, and we could move to a profitable business, cash generative, virtually immediately. But that obviously we've got to secure the customers that we've got in the pipeline. We've got about 20 customers in the pipeline, but we have been funded by our investors with a view that we should be looking to get a real major shareholder return and not keep the business narrow, but look for some major opportunities, which is obviously what we're doing.